continue to enjoy your lunch, and we will continue with our program. At this time, I am pleased to welcome Dr. Nowinski. Let's give her a big round of applause.
All right, thank you very much, Miranda, and thank you to Carol and Dave for the invitation to be here to speak with you all today. So, as you know, Carol and Miranda both alluded to when they were talking earlier, I think the first thing that most people think of when they hear metabolism is food. And you know, we all just enjoyed this wonderful lunch. Maybe you think about that, or maybe you think about how you can somehow snag a second dessert plate. Miranda just offered to watch mine so Dave doesn't eat it. So maybe you're trying out a new diet, or maybe you've tried every diet fad and none of them have worked for you. We're certainly bombarded with images in the media every day claiming to be able to hack or boost our metabolism, from diets and workouts to supplements and even devices now, all designed to help us to lose weight. And I think you probably all have a sense of this. Are we very good at this? Like, what do you guys think? I see some shaking heads. I want to hear you. Are we good at this? No. Because even when diets do work, we're even worse at keeping the weight off. And we're really bad at predicting what's going to work for any individual person. So how do we develop better strategies to do that? And how do we tailor those strategies to individual people? And why is it so important to do so anyway? So all of these cultural references to metabolism are talking about whole body metabolism, and that's what you see depicted here, where we take in oxygen and nutrients and water, and our bodies either burn those nutrients to produce energy, so we can walk around, keep our bodies warm, or we convert them into storage molecules, often in the form of fat tissue, or we excrete them as waste. And I think we're all pretty familiar with thinking about what whole body metabolism is, at least on some level, because we're all encountered with these choices and messages about diet and exercise constantly, every day. But what are the main factors that drive whole body metabolism, that determine whether that cheeseburger gets burned off as energy or gets stored around your midsection? In large part, these whole body decisions or trends are driven by cellular metabolism, by which pathways the nutrients take in the individual liver and muscle and fat cells in our bodies. So when I think of metabolism, I think of this definition. Metabolism is the series of chemical reactions that take place in cells to sustain life. And I think of this much more molecular definition, and that's what my lab studies. The thing is, there are a lot of chemical reactions that those nutrients can take. So say you ate some sugar. You know, sugar is going to enter your cell through the pink pathway up here, but then it has a decision. It can go through the green pathway, which is burning fats. It can go through the blue pathway, which is actually building DNA and proteins. Or it can go through the yellow pathway, which is burning for energy production. And so this is why cellular metabolism is such an active area of research, is because we're still mapping many of these pathways, and even more than that, we're figuring out how nutrients undergo these chemical reactions and how they decide which of those pathways they're going to take, what factors push nutrients in one direction or the other. And in part, one way that cells control and organize these reactions is via compartmentalization. So the main compartment that cells use to do the desirable burning for energy metabolic reactions is called the mitochondria. And you've probably all heard this phrase, mitochondria are the powerhouse of the cell. And that's because mitochondria are well known for their ability to take oxygen and nutrients and metabolize them to form ATP, which is the molecule that we as scientists think of as our cellular energy currency, because cells can use ATP to power things like muscle contraction. But cells have to balance supply and demand. They have to decide which nutrients are available that they can use to fuel these reactions. And they have to meet those reactions to the outputs that we need. Like, did you just run a marathon, or have you been sitting on the couch all day? And there are a lot of factors that are going to influence how that coordination takes place and which chemical reactions the cell or the mitochondria decides to use. So we started out by talking about diet, but as we've been saying, that's only one piece of the puzzle. And which metabolic pathways are available or preferred in our cells can be determined by factors including age, biological sex, fitness, pregnancy, illness, obesity, genetics, and even indirectly via external factors like socioeconomic status and stress levels. So there are studies that have shown that these environmental factors can all affect those decisions your body makes about what to do with nutrients when you eat. So this is the question that my lab is interested in. How do mitochondria determine what they are doing at each individual moment in time? What are the control mechanisms that regulate mitochondrial function, especially in the face of change, right? We don't all do the same thing every day. It might feel like that sometimes. But actually, sometimes you get up and you go to work and you have a lovely salad bar in the cafe that you can get lunch from. And sometimes it's Saturday and you go to McDonald's. And the reason we're interested in this question is that improper mitochondrial function is increasingly implicated in a whole host of pathologies. And these range from rare genetic disorders that we call inborn errors of metabolism to physiological aging to a whole host of age-related diseases like cancer, heart failure, Parkinson's disease. And in many of these diseases, the mitochondria aren't broken exactly, but it's more like cells just stop doing this balancing act correctly. And the balance shifts towards the mitochondria burning fewer nutrients for energy production and shifts more towards those building and storage type reactions. And there's mounting evidence in the scientific community that if we can shift mitochondrial function back towards that energy burning role, we can improve disease outcomes. So it's really not all just about losing weight, but targeting metabolism in a bunch of these other disease settings could be a useful adjunct therapy to help combat diseases like cancer and Parkinson's. So our goal is to better understand how mitochondrial function is controlled in normal cells so that we can then ask why and how that regulation is broken in disease settings and come up with ways to restore proper mitochondrial operation. And I'm going to tell you a little bit today about how we do that. But to understand that, let's look a bit closer at the machinery that mitochondria use to make that cellular energy molecule, ATP. So let's zoom in. On their inner membrane, mitochondria have this series of large protein complexes called the electron transport chain. These complexes are like little molecular machines. They accept electrons from the breakdown metabolic reactions that happen in the mitochondria. And those electrons flow down the chain like an assembly line until they get to oxygen. Oxygen is the ultimate electron acceptor in our bodies, and this is actually the part of our cells that breathes and consumes oxygen. And that flow of electrons lets these big molecular machines pump another charged molecule, protons, outside that inner mitochondrial membrane. What that does, that separation of charge across a membrane, literally sets up a molecular battery inside your cells. The cell can use that battery via the last complex in the chain, which lets the protons back in to make ATP. And in that way, you can think of the goal of the electron transport chain being to charge the battery. When the ETC is not plugged in, the battery's low. But when you plug in those metabolic reactions, you charge up the cellular battery. Now, here's the thing. This ETC charger, it's not just a cord. It's an assembly line. And each piece in this assembly line has to process and hand the electron off to the next machine. So I'm going to show you a video clip that demonstrates what happens if the flow of electrons through this molecular assembly line changes. And in this video, you can think of Lucy and her coworker as these electron transport chain complexes, and think of the chocolate as electrons. Oops. Yeah, you already know what's coming.
All right, girls. Now, this is your last chance. If one piece of candy gets past you and into the packing room unwrapped, you're fired.
Yes, ma'am.
Let her roll!
Oh, this is easier. Yeah, we can handle this, okay? Listen, Ethel, I think this... I think we're fighting a losing game. Here she comes.
All right. So again, if you think of the chocolate in the video as the electrons, when they started flowing too fast, many of them didn't end up where they were supposed to. They didn't charge the battery, and worse, they ended up in other places that we don't normally want electrons to be. Lucy could have really used a mechanism to check in and either slow down the chocolate flow or to call more workers to be able to keep up. So in the same way, the mitochondria needs ways to check in with the pathways that are supplying electrons to the chain, and then adjust how many electron chains the mitochondria has. And in my lab, that's what we study, how mitochondria do just that, either turn up or down the flow of electrons or build more of these complexes to be able to keep up. So how do we do it? How do we study mitochondrial function in cells? One tool that we use is a pair of fluorescent dyes that take advantage of that electrical charge separation that the electron transport chain makes across the mitochondrial membrane. So the dyes are called mitotracker green and mitotracker red, and we can put them on cells, and they're attracted to the mitochondria so that we can look at them under high-resolution microscopy and see what the mitochondria look like and how bright they are. The first dye, mitotracker green, it stains all the mitochondria, whether they're functional or not. So when we see less of it, it means the cell has fewer mitochondria in it. The second one, mitotracker red, it only stains functional mitochondria. So when the electron transport chain is not running, we'll see reduced or even no red signal at all. And this is analogous to what mitochondria look like in a lot of these pathologies I mentioned earlier, like diabetes or cancer. So you can think of it like a fluorescent open sign. When the electron transport chain is not working, the sign turns off. And so we use these dyes to look at cells from kids with a rare neurodegenerative disorder called MEPAN syndrome. And we didn't really know what was wrong in the cells from children with MEPAN syndrome before we did this experiment. But based on what we knew about the genetic mutations that these children have, we hypothesized that their mitochondrial health and function was probably disrupted. So here's what we see. In normal control cells, we have lots of green mitochondria, and they're nice and healthy, bright red, and working. In contrast, while the MEPAN cells still have lots of mitochondria that you can see in that green channel, their mitochondria are not lit up red. Their open sign is off. So we knew that the electron transport chain in these cells was not working. And this tells us something about how this mutation is causing disease in kids with MEPAN neurodegenerative disorder. But it also tells us something about how the gene mutated in this disorder is working in normal cells too, and that can inform our studies in other contexts. So to summarize, I hope what you take away from today is that many factors beyond diet affect cellular metabolism. Yes, we control what we eat, but our cells also have choices about what to do with those nutrients once they enter the cell. Mitochondria are metabolic hubs in cells, and their activity requires careful regulation, and they're a big part of that decision that cells have to make. In many diseases, our mitochondria don't operate properly, and this is a potential thing that we can target for treatment in the future. So by studying pathways that control mitochondrial function, we hope to develop strategies to target mitochondrial metabolism in disease. I have to end by saying thank you to the people at the Institute and in my lab who have done the work. So this is my lab this summer, and I want to be totally remiss not to thank the VII operations and course teams who really are an integral part of what we do every day, our many collaborators other places, and of course the patients and their families, as well as our funding sources. So I'm really interested to hear what questions you have, and let's have some good discussion. Right.
We will now open it up for questions, and we have some friends around the room with microphones. If you have a question, go ahead and raise your hand, and we will come around and bring the mic to you, and then you can ask that question. I'll get things started. I know that you are the mother of 2 young kids, and I would like to know if you're doing anything different based on the research you're doing as you feed your kids, as you have them involved in sports or other activities, knowing what you know, and what can you share with us on raising our kids to maybe have a better metabolism?
Oh, that's a great question. You know, I'd like to say, first off, my daughters are 4 and 7, and small kids are going to eat what they want to eat.
True.
So you only have so much control there. But I'm a big fan of the idea of everything in moderation, based on what I know about how our cells work and the metabolic pathways that we have. I think too much of anything is likely not to be very good. And on the flip side of that, I also think restrictive dieting or depriving yourself of things is likely not to be good or easy to maintain anyway either. And so I try to teach that to my girls. You know, yeah, have the Halloween candy, but maybe don't eat it all at once. And we try and really just make that the mantra in our household is eat a variety of foods, eat a healthy but varied diet, and don't eat in excess, just, you know, everything in moderation is really my favorite mantra.
Good answer, thank you.
Thanks.
Who has a question? Right here.
Are there specific nutrients that will help that mitochondria along to produce the energy?
So actually, most of the nutrients that feed mitochondria are really, we call them central carbon metabolism. So there are things like amino acids that come from proteins that we break down when we eat proteins, but they also come, you know, sugar digestive pathways can feed into mitochondrial metabolism, and fatty acid oxidation happens in the mitochondria as well. And so it's really about how the cell is deciding as those things come in, whether to fuel them, to feed them into the mitochondria or to feed them into more kind of like storage compartments and things like that. And you definitely can do things to help promote things going into the mitochondria. So one example of that is ketogenic diets, actually. So ketogenic diets really help promote mitochondrial oxidation of fats and, you know, less of those storage reactions. Mitochondria like sugar, so when you have sugar, they tend to store the fat and metabolize the sugar. On the flip side, though, we really don't know the long-term effects of doing ketogenic diet for a long time because it's going to affect other metabolic pathways like cholesterol biosynthesis. And so I'm not a clinician. I am not recommending any diet to anyone here today, and I want to be very, very clear about that. But I think that's why research in this area is so important so that we can understand these questions better and come up with better answers. So it may feel a little unsatisfying right now. I hope you didn't come to lunch saying, like, oh, this one has it. I'm going to go home and know exactly what to eat. I think the point is that we can't answer that question well, and that's why we need more research in this area.
Yes.
Hi, thanks. Great talk. So, one of the things we know about becoming overweight and obese is that we don't go to sleep one night weighing 95 pounds and wake up 195 pounds the next morning. And so this sort of gradual progression breeds in mitochondrial dysfunction. So that's part of the reason we rebound after we lose weight is we have now these dysfunctional mitochondria. We've sort of disrupted cellular energy dynamics. And is there a way then that we can start to think of how we can make those mitochondria healthy again so that we can sort of maybe combat this continual rebounding and weight gain?
Yeah, that's a really interesting question. So there are mechanisms that our cells have to be able to turn over diseased or damaged mitochondria as well. And so the question you asked is really, could we tap into some of those mechanisms to clear the damaged ones better? That's an active area of research, absolutely. I don't think we have a good way to do that yet, but it's a goal that there are labs working on, for sure. Yeah.
Yes.
Hi.
Hi.
How much is our metabolism related to our mom and dad? And
genetic.
are we what we eat?
Great questions.
I'm going to answer your second question first. So on a very basic level, yes, you are what you eat. Every cell that your body makes comes from the nutrients that you take in and how the cell is able to reorganize those chemical nutrients to make the molecules that make new cells and make your cells. So on a very basic chemical level, yes. But the reason that the title today is You Are More Than What You Eat is that two of you sitting next to each other aren't necessarily going to do the same thing with those same nutrients. And so that gets to the first part of your question, how much is it our mom and dad? I think genetics is actually a huge component of it. And that's the most common way that we study these things in the lab is actually looking at different genetic variations in metabolic pathways and looking at how they affect metabolism or how they affect these decision-making processes. I talked about MEPAN syndrome because it's a really clear example of somewhere that we use kind of the exception to study the rule. We study kids with this rare genetic disorder because it helps us understand how cells are working in normal people every day. And you just have a different flavor of that gene that happens to drive disease in those patients. So I think genetics is a huge factor, but it's not the only factor. And I think what it really is where you end up in terms of health and disease sits at the center of that gene and environment interaction. And in this case, your environment is your diet, right? So, really, there's things you can't change. You were born to the mom and dad you were born with. You can change your diet. And it's really the best you can do is sort of understanding and keeping track of what works well for you and what doesn't work for you.
Yes.
Hi. I lost a brother and a niece to mitochondrial disease. They were diagnosed, both of them, like around 26 years old. And it affected them brain function. Do you do much study with that? And is it rare that it hit that late?
Yeah, so we work a lot. Actually, one of my funders that was on my acknowledgement slide is United Mitochondrial Disease Foundation. I'm so sorry to hear about your family members. Mitochondrial disease is an incredibly difficult type of disease to diagnose and treat because it presents in patients very, very heterogeneously. Sometimes babies are born and it's caught very, very early in more severe forms of disease. And sometimes things don't manifest until adolescence or even adulthood. And one thing that we don't understand very well is where that variability comes from, because it is more variable than a lot of other types of inborn errors in metabolism. So MEPAN syndrome actually is classified as mitochondrial disease. We work with a few other mitochondrial disease models in the lab as well. And a lot of the time, what's also difficult with mitochondrial disease is that patients might be the only one with their specific mutation that's been characterized or sequenced or worked on. And so we, in many of these cases for these rare genetic disorders, we have no standard of care for those patients. And the treatments and therapies we have available are really more aimed at treating the symptoms than they are at treating the root cause of disease. So it's a very dire situation for those patients, and it's something that I hope to be able to impact as my research moves forward. I'm not going to answer it for every patient with mitochondrial disease because there are just so many diverse flavors, I guess, if you will. But being a parent myself, hearing your child has a disease that we have no idea how to treat would be one of the worst things I could imagine. And so I think it's a really important point because rare diseases like mitochondrial disease are actually overall very common. 1 in 8 people has something that's classified as a rare disease or disorder. It's just that there are so many of them. And so how do we begin to tackle that problem? It's really difficult.
Is there a tie-in between what we're seeing with drugs like Ozempic or Mounjaro with metabolism?
Yes. So Ozempic and Mounjaro, these GLP-1 agonists, have been really great drugs. They're really game changers for helping people with diabetes and obesity to get their weight back down, to be able to work with other medications and other doctors and things. So they've really been almost like miracle drugs in that field. And we're starting to explore them in other avenues too for patients with things like cancer and addiction. They work through hormonal mechanisms. So a lot of how our bodies determine whether we're hungry, whether we're full and satiated, is through hormonal signaling. And so that's a little different than what my lab works on, which is what we do with those nutrients once we consume them. But while I think that those drugs are wonderful and game-changing, we know now, as we're starting to see, that as patients come off those drugs, they gain a lot of the weight back. And they're also losing large amounts of lean mass while they're on the drugs as well. So often, it's not uncommon for a patient who's been on a GLP-1 agonist for a year to lose up to 30% of their muscle mass as well, which we would not consider a desirable outcome. And so I think that those drugs are wonderful and they're game changers, but I don't think they're the end-all be-all for metabolism research. I think that we still have more to understand. And so I think that I'm grateful that they're now a new tool we have in our arsenal, but we still need better understanding of other things moving forward too.
My question is that always we hear it's not about just the food, it's also about working out. For someone that doesn't like to work out, what do you think about intermittent fasting? And I saw results on my own kid that he thought his metabolism is not working right, seeing his friends eating a full whole pizza while they are so skinny, and he barely eats and he's not skinny.
Yeah, that's a great question. You will be happy to know that there are studies that show that if you exercise mice down to the same weight as calorie-restricted mice, so mice who have had their food intake restricted and are lean because of that, the exercise doesn't confer the same benefit as not eating as much. And that's probably because of that effect that we saw in the Lucy video where those electrons slip off all over the place. If you're burning more, those electrons tend to slip because our cells can't keep up. And so there's not the same thing as not putting calories into the circulation as burning more calories. The second part of your question was about intermittent fasting, though. And I think that research is really, really interesting because how do our circadian rhythms really interact with these metabolic pathways, and how does the timing of when we eat affect how we use the energy? And I think that the literature there is actually really quite good, that it is good to do intermittent fasting. It is helpful. Our bodies are more tuned to this. We're not supposed to be eating 24 hours a day. Similar from studies in mice. Mice are usually nocturnal, actually. Not a lot of people know that. So they'll eat a lot of their food during the dark cycle at night. But if you take it away during that dark cycle and give them the food during the day, during the light cycle instead, they'll gain weight, eating the same amount of food just at the wrong time of day when they're not supposed to. And so this is actually kind of bad for me because I'm a big breakfast skipper and I eat late in the day often. But really what you should do is try and time your eating to earlier in the day and not overnight. But again, not a clinician, not recommending diets. So.
It's good to know. Good to know.
Yeah.
Sara, thank you for your input. A person with your skills have lots of opportunities. If I recall, you spent time in Texas and Utah, but what attracted you to the Van Andel Institute, let alone West Michigan?
Good question.
So I grew up in Sterling Heights, actually. And so coming back to Michigan was a little bit like coming home. But I had other options in the Midwest, too, that I was considering. And what really brought me here to the Van Andel Institute was two things. The first was having a dedicated metabolism department. We have a department with 6 labs now, hopefully looking to grow in the next several years. And having that kind of community that you can discuss research with, bounce ideas off of, share resources, I thought was really valuable. It's a little bit more of a team approach to science, even though we all have our specific research areas and do different things. I know the value of community, and so I really valued that focus that existed here. Versus most other places, a metabolism lab would be in a large biochemistry department that's doing lots of other disparate things. The other thing that brought me to the Van Andel Institute was really the world-class resources that are here. The support here is phenomenal. The core facilities are really, really wonderful. The operations staff take care of everything for you so that as a scientist, I can really focus on the science and mentoring the young researchers in my lab, helping them figure out problems, and that's really where I wanted my focus to be.
Sara, over here. What is the correlation between metabolism and sleep quality and quantity?
Gosh, you guys are with the sleep. I don't know that I know what the exact correlation is. Sleep really isn't my area of expertise. I do know that there's research showing that both too much sleep and too little sleep is bad. But what I don't know are the sort of, like, what are the connections between metabolism and sleep? Yeah, great question. I think it's an area that I know is an active area of research, too. So, thanks.
Good. We have time for one more. Yes.
Okay. Hi. Quick question on, I used to ascribe to all food is equal as long as you balance, but really understanding more about metabolic syndromes and diseases tied to high fructose corn syrup and seed oils, all the highly processed foods, and then how our bodies react to that, and then the inflammation that we end up carrying because of the processed foods. So, really understanding like the best foods, and again, timing too, I think is important. But what is the connection between mitochondrial health and inflammation?
Yeah, great question. And actually, you hit on, as you were talking, I was like, oh, that's what Evan Lien's lab studies. Oh, that's what Rusty Jones and Connie Krozik's lab studies. So we have labs in the building that are actively asking a lot of those questions, looking at different types of polyunsaturated and monounsaturated fatty acids, which are present in palm oil or corn oil or avocado oil at different percentages. And what effects does that have on metabolism and on diseases like cancer? As well as, exactly what you said, inflammation and metabolism, we know, have this really complicated relationship, for sure. I don't know that I can answer in a good, succinct, direct way, other than to say there's a lot of research in this area, and we don't know the answers well yet. But I think one thing that I can say is that my lab is collaborating with the Jones lab to look at if we break mitochondria, if we make mitochondrial dysfunctional, then what happens to T cells and their responses to infection and inflammation and things like that? And what it seems like we're seeing is that the T cells look like they're entering this state that we call exhausted. And it's a state that you would see like after a chronic infection when the T cells have been working really hard and now they're not really working that well anymore. So just breaking their mitochondria when they haven't experienced an infection that's chronic that would normally push them into this state, we're seeing them look like they're in this exhausted state. And so really understanding and describing how that happens is an active goal for us right now. But yeah, great ideas as well.
Is she phenomenal or what? Let's give her a round of applause. Thank you.
Thank you.
You know, I love how charming you are, how approachable, and how brilliant. And you know what? The Van Andel Institute is full of scientists just like her. So on that note, I want to thank you, Carol, for bringing one of the scientists to the spotlight so we can see the good work being done here. Thank you for hosting this lunch, Carol. Thank you so much for sharing, Dr. Sara. We so appreciate it. Today's exciting because you have the opportunity to get involved. And if you are here and you're listening and you're like, I want to learn more or I want to support the good work Dr. Sara and her team are doing, we have a great opportunity. Inside your program, inside you will see a QR code right underneath her beautiful photo, and you can go ahead and scan that and make the donation directly that way. You can also find out about all of the activities happening at the Institute at vai.org or on our social platforms, but it's just another way for you to plug in to continue to do these kinds of opportunities to help us all. We have a great public lecture series coming up in December that, again, gets into this topic on environmental impact on our health. We sure would invite you to join us for that as well. We want to thank our sponsors today for stepping up to the plate and joining us to have this incredible dialogue, and I want to thank you all for giving your time today. Again, thank you, Carol, and thank you, Dr. Sara, and I hope you have an amazing afternoon. Thank you.